Okay, so welcome back to this next video in which we are discussing the Wnt beta-catenin pathway, which is also known as the canonical Wnt pathway. Okay, right, so we have now discussed the frizzled receptors and the LRP receptors. Okay, we now want to discuss how Wnt is going to bind to them, and then how they're going to dimerize, and then what downstream pathway that is going to trigger, specifically looking at the canonical Wnt pathway. Okay, right, the Wnt molecule, and we've seen that there are 19 different genes for Wnt molecules within mammals, is going to bind to the extracellular domain of a frizzled receptor. So let's draw this now down here. So here is our cell membrane, okay, and then we've got some frizzled receptor here. Here's the extracellular domain. Then we've got our 7 membranes spanning alpha helices of this frizzled receptor down here, ending with the carboxylic acid terminus intracellularly here. Okay, and here's the amino terminus. Now we've got our Wnt molecule here. Okay, like so. Now, this is a protein, of course. Now, the Wnt molecule is going to bind to it's going to bind to the LRP protein now as well. Now there are 2 different sites on the LRP protein that Wnt proteins can bind to. Okay, so let me now show you these 2 different sites. You remember I told you that there are loads of different Wnt proteins, and these do differ in their primary sequence of amino acids. Well, the different Wnt proteins are going to bind to the LRP proteins at different sites. Okay, so some Wnt proteins like to bind between the P1 and the P2 beta propellers. So they'll bind to a portion of the P1 beta propeller, and they'll also bind to a portion of the P2 beta propeller. Okay, whereas other Wnt proteins prefer to bind between the P3 and the P4 beta propellers. So they'll bind to a portion of P3, and they'll also bind to a portion of P4. Okay, so there are those 2 different Wnt binding sites, and which one is used will depend on which Wnt molecule you are using, basically, to bind to these receptors. Okay, so these are Wnt binding domains here. Now, I don't think I'll be able to squeeze my LRP protein in so that I have it bind this Wnt molecule here binding to the P1, P2 portion here. So instead what I'm going to show is it binding to this junction between P3 and P4. Okay, because I'll actually be able to fit that in. So, here is my P4 beta propeller here, shown as this hexagon. Then we've got my P3 beta propeller here, and the Wnt is binding to both of them. Okay, then we've got our P2 beta propeller up here, and then the P1 beta propeller above that. Okay, then you have the amino terminus even prior to that. Then spanning the membrane you have your single membrane spanning alpha helix, followed by your carboxylic acid terminus intracellularly. Okay, so let's now colour in these beta propellers in blue here. Okay, so that we've got nice parallels between our 2 drawings. Okay, so this Wnt molecule is specifically binding to both the P3 and the P4 beta propellers, and that linker between those 2 beta propellers. Okay, right, so we now have this dimer of our frizzled receptor here, which I'll abbreviate as FZ, and then our LRP receptor here, which is either LRP5 or it's LRP6. Okay, this now is going to activate the beta-catenin pathway. Now, when discussing the Wnt beta-catenin pathway, it is better to firstly discuss what the Wnt beta-catenin pathway is doing before you actually activate the receptor like this. Okay, so the Wnt beta-catenin pathway has basically 2 states. It has an off state, okay, and it has an on state. Now the off state is the state that the Wnt beta-catenin pathway within the cell will be in when there is no Wnt around and there is no dimerization of the frizzled receptors with the LRP5/6 receptors. Okay, so it's better to firstly discuss the off state and then we'll come on to the on state after we've discussed the off state, so that we can then discuss what's going to change basically. So we're going to start by discussing what is happening within the cell to beta-catenin before the Wnt arrives and dimerizes the frizzled receptor with the LRP5/6 receptor. Okay, so let's now discuss this. Okay, so we need to discuss something called the beta-catenin destruction complex, okay, which is going to be involved in making sure that in this off state, no beta-catenin survives, okay? So firstly, let's just introduce beta-catenin. So beta-catenin is a protein, okay, that's going to function as something known as a transcriptional activator, and we'll discuss what a transcriptional activator is much later. Okay, but for now we'll just represent our beta-catenin protein. Right, so here's the amino terminus of our beta-catenin protein. Then this is the bulk of the beta-catenin protein, and then it's got a carboxylic acid terminus at the other side. Okay, so we'll colour this in green here. Okay, right. Now usually within the cell you don't have any of this beta-catenin. Even though you're making it continuously gets destroyed. Okay, and we now want to see which proteins are involved in destroying this beta-catenin usually. Okay, so usually you have a complex of proteins which is known as the beta-catenin destruction complex, which is involved in destroying the beta-catenin. Okay, and I now want to discuss which proteins are going to be complexed together to make this beta-catenin destruction complex. Okay, so firstly there are two what are known as scaffold proteins involved in this beta-catenin complex. Now scaffold proteins are just proteins which bind to other proteins and hold them together basically. Okay, so the first scaffold protein we're going to discuss is a protein known as axin. Okay, so we'll colour in axin here in orange. So this orange rectangle, this is representing axin, and it's what's known as a scaffold protein, just meaning a protein that binds to loads of other proteins and holds them all together. Okay, right. Now, firstly, what I want to discuss is then it binds to another protein, which is also another scaffold protein. Okay, so here is another scaffold protein that the axin itself is bound to. Okay, and this second scaffold protein is known as the APC scaffold protein. Now, what does APC stand for? Well, it stands for Adenomatosis polyposis coli. Okay, so it's named after the disease, which is a hereditary predisposition to colorectal cancer that was found to be associated with mutations in this protein. Okay, so APC stands for Adenomatosis polyposis coli. Okay, right, so the A is for adenomatous, the P is for polyposis, and then the C is for coli. So this is the full name for APC. And APC is another scaffold protein, so it's just a protein which binds to other proteins and holds them together. Okay, right. What else then is going to bind onto here? Well, we're now going to get some enzymes binding here. So the first enzyme I want to discuss is something known as glycogen synthase kinase 3 beta, which is often abbreviated to GSK 3 beta, like so. Okay, so what does that stand for? That stands for, the G is for glycogen, okay, the S is for synthase, okay, the K is for kinase, and then it's just glycogen synthase kinase 3 beta. Okay, so this is usually abbreviated to GSK 3 beta. Okay, right, so this binds to both the axin and the adenomatous polyposis coli here. So we'll have this in green. Okay, like so. So that's glycogen synthase kinase 3 beta. Now, this is a kinase enzyme, which is why I've drawn it with that active site there in the classical way of drawing an enzyme. Okay, and the reaction it's going to catalyze is the phosphorylation of serine and threonine residues within proteins. So I just want to discuss what a serine threonine kinase is, because that's what glycogen synthase kinase is. It's a serine threonine kinase. So let's just take a little bit of time out to discuss what serine threonine kinase enzymes actually are. So we'll start by discussing what serine and threonine residues within proteins are, and then we'll discuss what a kinase enzyme is going to do to these serine and threonine residues within proteins. Okay, right, so let's start with what a serine residue is then. Okay, so we're going to draw it as a residue. Okay, and when I say we're going to draw an amino acid residue, what that means is we're going to draw the amino acid as though it's within a polypeptide. Okay, so we're going to draw the amino and carboxylic acid groups as though they're bound to other amino acids by peptide links. So here's the amino group of our amino acid residue, and I've drawn it as though it's bound to the carboxylic acid group of the amino acid prior to it. Then we have the alpha carbon with a hydrogen coming off. Okay, then we have the carboxylic acid group here. Okay, and that's the core amino acid residue structure now. The R group in the case of serine residues consists of a methylene group, shown here, then with an alcohol group coming off that. Okay, so this is the structure then of a serine amino acid residue here. Okay, right. Now let's see the structure of threonine amino acid residues. Now, threonine is very similar to serine. So here's the amino group. Again, bound with a bond which will be going to the carboxylic acid group of the amino acid prior to it. Here's an alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group again with a bond which will be going to the amino group of the amino acid after it. Okay, and then the R group of a threonine residue consists of a very similar structure to serine. You've got a carbon with an alcohol group coming off it, and then this carbon also has a hydrogen coming off it and a methyl group coming off it. Okay, and this then is the structure of a threonine amino acid residue. Okay, so both of these residues can be phosphorylated by serine threonine kinase enzymes on their alcohol groups. Okay, so let me show you what this means. So if we take a phosphate group here, a phosphate group consists of a phosphorus atom double bound to an oxygen atom with an alcohol group coming off it, and then with another alcohol group coming off it this side, and then a single bond to an oxygen atom with a negative charge coming off it like so. Now this is actually a free molecule known as an inorganic phosphate. Okay, to create a phosphate group, we're going to attach this onto the threonine residue here, or the serine residue. It's completely analogous for the serine residue as well. Now, in reality, serine threonine kinases are not going to take inorganic phosphates from the cytoplasm and just add them onto serine and threonine residues. Instead, what they do is they take them from adenosine triphosphate molecules. Okay, so they take ATP, chop off the terminal phosphate group on that, and transfer it onto the alcohol group of the serine or the threonine residue. However, binding an inorganic phosphate onto a serine or threonine residue will highlight the principle of what's going to happen when we phosphorylate a serine or a threonine residue. Okay, right. So, basically, the reason that you can attach inorganic phosphates, or indeed phosphate groups, onto alcohol groups is because if we look at this group here, which I'm now circling in turquoise, okay, this phosphorus atom double bound to an oxygen atom with an alcohol group coming off, what does that look very similar to? well, it looks very similar to a carboxylic acid group. In fact, if we replace that phosphorus atom with a carbon atom, that would be a carboxylic acid group. Now, carboxylic acid groups can react with alcohol groups via ester links. Okay, so in fact, these carboxylic acid-like groups that we have within phosphate molecules, like so, can actually react very similarly to if they were carboxylic acid groups with alcohol groups. Okay, so what's going to happen is you're going to take the alcohol group off the phosphorus atom. You're going to take the hydrogen off the alcohol group. You will combine that alcohol group with the hydrogen to produce water. Okay, that's why this is a condensation reaction. And you will then bind the oxygen atom of the serine or threonine residue's alcohol group to the phosphorus atom of the phosphate group here. And that link that we've created here is what's known as a phosphoester link, drawing the parallels between this kind of a link and an ester link between a carboxylic acid group and an alcohol group. Okay, so this is a phosphoester link. Right, so that's what's going to happen when we phosphorylate serine and threonine residues. You're going to end up with phosphate groups attached via phosphoester links onto serine and threonine residues. Okay, right, so that's what these glycogen synthase kinase 3 beta enzymes are going to do. And we've got one of these within this beta-catenin destruction complex. Okay, now there's a final protein that we want to put in here. Okay, and I'll stick this in here. Okay, so this final protein is going to be another serine threonine kinase enzyme. And this is what's known as casein kinase 1 alpha. Okay, and we'll colour in casein kinase 1 alpha in pink here. Okay, so CK1 alpha stands for, if I write it down here, it stands for casein, that's the C, then the K is for kinase, and then it's casein kinase, specifically 1 alpha. Okay, so this is for short is CK1 alpha. Now, you're going to have a casein kinase 1 alpha molecule attached to the axin scaffold protein within our beta-catenin destruction complex. And this now is the beta-catenin destruction complex fully assembled. Okay, this is now going to start the process of destroying beta-catenin molecules. Okay, so we are currently discussing the off state of the Wnt beta-catenin pathway. So there is no Wnt in the extracellular fluid at the moment. There is no activation of the frizzled receptors or the LRP5/6 receptors. Okay, so at the moment the beta-catenin destruction complex is therefore fully operational. So we've got these things assembled. We're going to break them down when the Wnt actually arrives in the extracellular fluid and activates the frizzled and LRP5/6 receptors. Okay, but for now, the beta-catenin destruction complex gets to have its fun. And what's going to happen is it's going to bind a molecule of beta-catenin in here. Okay, so I'll show this like so. So here comes a molecule of beta-catenin, and unfortunately I've drawn it in the same colour as I've shown glycogen synthase kinase. So I'm going to change the colour of beta-catenin now. It's going to be represented in red, okay, just so that I don't have two things in green on this picture. Okay, right, now the amino terminus will be this side, so here's the amino terminus, and then the carboxylic acid terminus will be on this side. Okay, right. So it's going to bind to the beta-catenin destruction complex, and then what's going to happen is our serine threonine kinase enzymes, which we've got here, which are the casein kinase 1 alpha enzyme and also our glycogen synthase kinase 3 beta enzyme, they are going to phosphorylate serine and threonine residues on this beta-catenin protein. Okay, so the story so far then is we've just produced this beta-catenin molecule, let's say. It's just been translated, it's just come out of a ribosome, now it's bound to our beta-catenin destruction complex, and now it's going to get broken down, but not directly by the beta-catenin destruction complex. Instead, what's going to happen is the beta-catenin destruction complex is going to label it up for degradation. It's going to put markers on it which say, okay, destroy this, okay? So these are going to be phosphate groups. So firstly, casein kinase 1 alpha is going to begin the phosphorylation. Okay, so it's going to add a phosphate group onto the beta-catenin molecule. Okay, so this is done by casein kinase 1 alpha, and this is important. The first phosphate group is always added on by casein kinase 1 alpha, and specifically we are going to phosphorylate a serine residue, and I even have the number for this, and this is quite important. It's serine 45. Okay, so you're going to phosphorylate serine at position 45. Okay, so the 45th amino acid in beta-catenin is a serine. We're going to phosphorylate that. Okay, so casein kinase 1 alpha begins the phosphorylation by phosphorylating a serine residue at position 45. Okay, right. Now, that's what's known as the priming phosphate group. Okay, so this primes the beta-catenin now to be phosphorylated by the glycogen synthase kinase 3 beta, and glycogen synthase kinase 3 beta is now going to phosphorylate 3 different phosphorylation sites, which are all upstream, okay, so closer to the amino terminus of the polypeptide than this phosphorylation site that was originally phosphorylated by casein kinase 1 alpha. So these 3 further phosphate groups here that we've added on to take us up to a total of 4, these were all done, all 3 of these were done by glycogen synthase kinase 3 beta. Okay, now what are the specific sites that are phosphorylated? Well, I've got the numbers and the residues again. Okay, so the one just upstream of the serine 45 is another serine. Oh, sorry, not a serine residue, a threonine residue there. Okay, so this one up here is going to be a threonine residue, and it's going to be threonine 41. Okay, so threonine 41. Then the one upstream of threonine 41 is going to be a serine residue, and that's going to be serine 37. Okay, and then finally the one upstream of serine 37 is going to be another serine, and that's going to be serine 33. Okay, so overall, these 4 phosphorylation sites are serine 33, serine 37, threonine 41, and then serine 45. Okay, so to review the entire process, casein kinase 1 alpha starts. It starts by adding a phosphate group onto serine 45. That gets the beta-catenin ready so that now glycogen synthase kinase 3 beta can phosphorylate threonine 41, serine 37, and serine 33, so that now the beta-catenin has these 4 phosphate groups on it. Okay, now what's going to happen is another protein is now going to bind to this phosphorylated beta-catenin. Okay, so let me draw this. So here now is our beta-catenin after it has been phosphorylated by the beta-catenin destruction complex. Okay, so now it's going to come off the beta-catenin destruction complex and it's going to bind to a different protein now that it's got these 4 phosphate groups added onto it. Okay, so here is our beta-catenin molecule in red here. Okay. Here are our 4 phosphate groups. This one is serine 45, this one is threonine 41, this one is serine 37, and this one is serine 33. And now another protein is going to come and bind to this. Okay, here it is, like so. And this protein is a protein known as beta-TRCP. Okay, so this is beta-TRCP. Okay, so let's colour in beta-TRCP in green here. And this is an enzyme which is capable of adding ubiquitin groups onto proteins. Okay, so what's going to happen now is that it's going to stick a ubiquitin group onto the beta-catenin molecule. Okay, so we'll stick it on here. So here is the beta-catenin molecule that's now got this ubiquitin group stuck onto it, and I'll colour this ubiquitin group in orange here. Now, ubiquitin is not what proteins want to get stuck onto them, okay? Ubiquitin is the ultimate marker for a protein to be destroyed, okay? So, the beta-catenin destruction complex phosphorylates beta-catenin at these 4 sites so that this other protein here, beta-TRCP, can bind to the beta-catenin, and then it will catalyze the addition of a ubiquitin group onto the beta-catenin. And by the way, ubiquitin groups are often just abbreviated as UB groups. Now, why do proteins not want to have ubiquitin groups added onto them? Well, because ubiquitin groups send you to the proteasome. Okay, so what is the proteasome? Well, the proteasome is a truly terrifying thing in the world of molecular biology. Okay, so if you're a protein, the proteasome is terrifying. So the proteasome is a tube, basically, and you've got a hollow channel through the middle of this tube. Now, proteins go in one side, okay, and then fragments of proteins come out the other side. Okay, so in the middle of this tube, you're going to have loads of enzymes which are going to break down the proteins. They're not quite going to break them down into amino acids, but they'll break them down into small fragments of amino acids. So, ubiquitin is the molecule which then binds to the entrance of the proteasome and results in a protein being fed into the proteasome. So what's now going to happen is this beta-catenin is going to bind to the entrance of the proteasome here, because the ubiquitin molecule will bind there, and then the beta-catenin is going to get fed through the proteasome and therefore is going to be destroyed by the proteasome. So that is the Wnt beta-catenin pathway in the off state. Okay, so before any Wnt molecules have arrived, before you've had stimulation of the frizzled receptors or the LRP5/6 receptors, at the moment, all of the beta-catenin that you are producing is going to be firstly phosphorylated by the beta-catenin destruction complex and then ubiquitinated by beta-TRCP and then destroyed. So no beta-catenin is getting to actually do anything at the moment. Okay, right. We will have a break there, and then in the next video what we'll now look at is how when Wnt arrives and activates the frizzled receptors and the LRP5/6 receptors, how this is going to stop the beta-catenin destruction complex from functioning and turn the Wnt beta-catenin pathway into the on state, which is the state where beta-catenin is actually going to build up within the cytoplasm of the cell.